phenotype of the parent, and then determine what two alleles are possible. And then you cross that by the other parent, and then you just match it up. So this organism is going to be homozygous dominant with a capital P, capital P. This guy right here is going to be big P, little p, so another heterozygous. This guy is also going to be heterozygous, big P, little p. And then this guy over here, he's going to be little p, little p. So this is how he observed that 3-to-1 phenotypic ratio. It's okay. Go ahead.
So in the top left, it's the dominant homozygous. The bottom right is the recessive homozygous.
Yes.
And the other two are the heterozygous.
Heterozygous,
Okay.
yeah. So these are both homozygous. All right. This one's just homozygous dominant, and this one's homozygous recessive.
Okay.
And then the two that have one of each allele, those are going to be the heterozygous.
Okay.
And so from this cross in the F2 generation, again, this is how he saw that 3-to-1 ratio. So he saw 3 purple flowers for 1 white flower. And so you should notice that even if you have a heterozygote, so the phenotype of these two plants are going to be the exact same. They're both going to appear purple, but the genotype is going to be different. In this case, this is going to be a big P, so a homozygous dominant. And in this case, this is going to be a heterozygote, so a big P, little p. So even though the genotypes are different, you can still have the same phenotype. And so we can compare, Mendel started off looking at the phenotypic ratio. And so the phenotypic ratio observed is 3-to-1, 3 purples and 1 white. Now the genotypic ratio, you end up with 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous. So in terms of a genetic ratio or a genotypic ratio, he saw a 1-to-2-to-1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And so the genotypic phenotype is a 1-to-2-to-1, while the phenotypic ratio is going to be a 3-to-1. So if it's a phenotypic, you're just basing it on what it looks like. But if you're looking at the genotype, you're actually looking at the different versions of the alleles that are present. Okay, so these are some terms that we've kind of already gone over at this point, but we'll go over them one more time. So when we're talking about the genotypes of these different types of plants or whatever organism we're talking about, they're going to be either homozygous or heterozygous. Homozygous means that it's going to have the same copy of the allele. So in other words, it's going to have either a capital P, or a lowercase p. So they're going to be the same. And so they can either be homozygous dominant or they can be homozygous recessive. If they're homozygous dominant, that means that both alleles are going to have the dominant allele present. And if they're homozygous recessive, that means that both chromosomes are going to have the recessive allele present. And then you can also have heterozygous, which is where you get 1 dominant allele and 1 recessive allele. And so in the case of the heterozygotes, as far as Mendelian genetics is concerned, the heterozygotes, because they contain that dominant allele, they're going to look exactly like the homozygous dominant, even though they are heterozygous themselves. Questions so far? All right, so this is comparing the genotypic to the phenotypic ratios that Mendel observed. So in his F2s, you know, he crossed the parents and he got the F1s, and then he crossed the F1s to one another to get the F2s, and this is what he saw. So again, he saw 3 purple flowers and 1 white flower, so a 3-to-1 phenotypic ratio, just based on what the flower looks like. And then when you actually compare the genotypes, you can see this 1-to-2-to-1 ratio. So 25% homozygous dominant, or big P, 50% heterozygous, or big P, little p, and 25% little p. or, the white flower. And so, again, just comparing the genotypic to the phenotypic ratios, just because the genotypes are different doesn't necessarily mean that the phenotypes are going to be different from one another. Okay, so we already talked about Mendel's law of segregation, saying that the two different versions of a gene are going to separate into different gametes during meiosis. From his research, Mendel also came up with the law of independent assortment. And so that law of independent assortment deals with how those homologous chromosomes line up with one another in metaphase 1 of meiosis 1. And so remember, we just said that depending on how those homologous chromosomes are arranged can dictate which gametes get put, which alleles get put into which gamete. And so if we look at the different combinations, remember we can have, so if we've got, mom's chromosome and dad's chromosome, depending on how these align, is going to dictate which alleles get distributed in which gametes. So they could be arranged like this. So this, just imagine this is the metaphase plate. Or they could be arranged with both of mom's chromosomes on one side and dad's on the other. So depending on how many chromosomes a cell has, that's going to give you increased genetic variation just based on the way that these homologous chromosomes are lined up in metaphase. And so from this, Mendel concluded that in metaphase 1 of meiosis 1, that the way that this pair of homologous chromosomes lines up is totally independent from this pair. So in other words, the way that each homologous chromosomes are arranged are totally random and totally independent of any other pairs of chromosomes. So in other words, just because this pair, ended up arranged like this doesn't mean that this guy's going to, change his arrangement, because it's all random. It's all by chance. So in other words, the way that these chromosomes align is totally independent from the way that any other set of homologous chromosomes are going to align. And that's what the law of independent assortment is. So if they assort independently, that means that you're going to end up eventually separating those different alleles of those gametes during metaphase 1. And so what we've been talking about have been these monohybrid crosses that are only looking at one particular trait. Mendel also did some research looking at two different traits. And so when you look at two different traits, we call this a dihybrid cross. And so di in Latin means two, and hybrid means we're still going to be crossing the F1 generations to one another to get the F2. It's just in a dihybrid cross, instead of only looking at one particular trait or gene, we're looking at two of them. And you can also do a trihybrid cross, where you're crossing F1s and you're trying to follow three different genes. But obviously the more genes you are throwing in there, the more complicated the genetics get. So as far as we're concerned, we're going to stop at a dihybrid cross. One other thing I did not mention, in the law of independent assortment, this is only going to be for genes that are on non-homologous chromosomes. And so those non-homologous chromosomes are basically going to be but they're going to assort independently. So if you look at, in other words, if you look at this chromosome right here, and there might be a trait right there, and then this is a totally different chromosome and a trait right here, it's saying that this trait and this trait are only going to be found in, well, it's going to be here and here as well. But basically, each version of this trait is going to be separated during meiosis 2. Okay, so Mendel also started looking at these dihybrid crosses, looking at two different traits instead of one. So one of the other studies he did was he again used these pea plants, but instead of using flower color, he used seed color and seed shape. And so what he did is, again, he took these true breeding, so these homozygous dominant or homozygous recessive pea plants. In the case of the dominant ones, he took a yellow colored with a round seed. So yellow is dominant and round is dominant compared to green and wrinkled. And so what he did is he took these, these parental crosses, these true breeding phenotypes, and he crossed them to one another and got the F1. And in the F1, he's going to see the dominant phenotype. So he's going to see the yellow and he's going to see the round seed. Then he took those F1s and he mated them together in this dihybrid cross, and what he noticed was a different phenotypic ratio. So instead of the 3-to-1 phenotypic ratio, he observed a 9 to 3 to 3 to 1 ratio. And so that 9 to 3 to 3 to 1 is going to show most of the dominant traits over the recessive traits. And so if you look at the Punnett square for determining what these offspring might look like, you now, instead of just having one allele, now you're looking at two different alleles. So in this example, since this is a dihybrid cross, he took a heterozygous F1 times a heterozygous F1, because all the F1s were heterozygous. So in this dihybrid cross, where you're crossing the F1s, you're crossing two heterozygotes to one another. And so if you look at the heterozygote genotype, you've got big Y, big R, little r. So in meiosis, you're going to get two different combinations of gametes. Either the big Y and the big R can come together, or the little y and the little r can come together. And so you can see the different arrangements right here. And same thing with the other one. There's only two different ways that those gametes can assort during meiosis. And so you end up crossing a big Y, big R to a little y, little r, and a big Y, big R to a little y, little r. Because the only possible arrangements of those gametes are going to produce that. So if we have a heterozygous right here, and you can only have two different combinations of alleles after meiosis. You can end up with big Y, big R, or you can end up with little y, little r. And so when you look at the outcome of this this dihybrid cross, he noticed that that you can get 3/4 yellow to the 25% green. But when you look at the actual results that he gets, he noticed that you can get actually more combinations of these gametes. So you can get YR, or homozygous dominant, and homozygous recessive, but you could also have the possibility of having big Y, little r. Just like there's a possibility of having little y, big R. And so in the dihybrid cross, you're always going to have 4 different genotypic possibilities of those gametes. And so you can see them all right here. You can either get big Y, big R, big Y, little r, little y, big R, or little r, little y. And so if you cross all those different possible combinations together, you're going to get 16 different offspring, or not necessarily 16 offspring, but you're going to get a ratio of 16 different possible genotypes. And of those genotypes, 9 out of 16 of them are going to have the dominant characteristics. So in other words, the yellow seed coat and the round seed color. And then he noticed that there were 2 kind of intermediate phenotypes. One that had one of the dominant traits and the other one was recessive, and vice versa. So in this case, the green and the round are going to be heterozygous for each trait. So in other words, it's going to show the dominant seed shape, but it's going to show the recessive seed color. And this guy is going to show the dominant color, but the recessive seed trait. And then the last guy, the 1 out of 16, he's going to be the double recessive. So he's going to be homozygous recessive for both traits, meaning he's going to be green and he's going to be wrinkled. So long story short, in these dihybrid crosses, you're going to end up, if they're exhibiting Mendelian genetics, you're going to end up with a 9-3 to 3-1, with the dominant phenotypes being more readily available compared to the recessive phenotypes or the recessive traits. All right, questions? I feel like I might have missed you here. Lost some of y'all. Y'all doing okay? All right, sweet. Okay. So all we've been talking about at this point is what happens in simple genetic crosses, but everything isn't as simple as we always make it out to be. So different genes can act differently compared to Mendelian genetics. So Mendelian genetics doesn't always apply to a situation. Sometimes it gets a little bit more complicated than that. And so these Mendelian inheritance patterns aren't always held up to 100% of the time. And so the reason why is because not all alleles are going to act in this totally dominant and totally recessive fashion. In other words, some alleles might not show the full amount of dominance compared to, say, the purple flower trait. In the purple flowers, as long as you had that capital P, that means you were going to be purple. Well, not all traits show that much dominance. Also, you can have a gene that has more than 2 alleles. So the only thing Mendel is looking at is when you have 2 opposing traits, white versus purple, brown versus wrinkled. Looks like we're in their lab for some reason. Give me a second.